Okay, let's get right into it. Welcome to this explainer. If you're a focused learner, currently staring down the barrel of an upcoming genetics and cell biology exam, you are in exactly the right place. Today we're taking a mountain of complex biological data and transforming it into a highly structured, memorable study guide. We're going to completely strip away the noise and focus purely on the core concepts, the evolutionary timelines, and the essential vocabulary you need to not just pass, but absolutely ace your test. So, here's our game plan for this session. First, course grading. Second, the epic origin of life. Third, the 3 life domains. Fourth, comparing cell types. And we'll wrap up with essential key terms.
Section 1. Course grading and expectations. Because before we shrink all the way down into the microscopic world, we really need to address how you're actually going to be evaluated in this course. Okay, let's dive into this breakdown. Your entire course has a maximum score of 60 points, but look really closely at how those points are distributed. Every single category is weighted equally at 10 points each. You've got 3 separate tests, a term paper, your final practical exam, and class attendance. So here's a massive strategy note for your studying. Class attendance is mathematically just as important as your final practical exam. Showing up is literally a 1/6 of your entire grade.
Section 2. The epic origin of life. Now that we've conquered the modern syllabus, let's travel back about 4 billion years to set the stage for cellular biology.
To truly understand the cells operating inside of you right now, we have to look at this chronological timeline. Around 4 billion years ago, Earth was this harsh, chaotic mix we called the primordial soup. But as we move forward to roughly 3.9 billion years ago, biological activity really started concentrating around alkaline hydrothermal vents deep down in the oceans. This extreme energy and crazy chemistry gave rise to the RNA world around 3.85 billion years ago, which is where the first self-replicating molecules emerged. Finally, by about 3.8 billion years ago, those molecules became enclosed in the very first cell-like bubble structures, and that right there is the dawn of biology.
Just picture this environment for a second. Deep, boiling, extreme, and totally toxic to almost everything we know today. Yet it was in these very alkaline hydrothermal vents that early extremophiles totally thrived. Specifically, aerobic heterotrophs managed to survive here by utilizing that intense heat and those wild chemical gradients. And amazingly, these incredibly resilient organisms, they are the ancient ancestors of the very first eukaryotes, which means they're literally the ancestors of us.
Section 3. The 3 life domains. Because from those first resilient bubbles of life deep in the boiling ocean, a massive evolutionary divergence took place.
And this brilliantly illustrates that split. Every single living thing you have ever interacted with or that has ever existed on this planet falls onto the structural tree. It's divided into 3 distinct domains: bacteria, archaea, and eukaryota. Understanding where specific organisms fall on this tree is foundational to literally everything else you'll do in cell biology. Now, to pass your exam, you can't just know the names of the domains, right? You need to know their specific biochemical traits. This breakdown is an absolute must-have for your test prep. Notice the patterns here. Archaea and bacteria both have circular chromosomes and both lack a nuclear envelope. But look at peptidoglycan in the cell wall. That is completely exclusive to bacteria. In membrane-bound organelles, that's the hallmark trait entirely unique to Eukarya. Seriously, memorize these distinct checkpoints. They're absolutely going to show up on your tests. Section 4. Comparing the cell types. While there are 3 domains, your course material primarily demands that you understand the 2 core cellular architectures that make them up. So, the crucial point here is grasping the clear, study-ready contrasts between these 2. The prokaryote is small, measuring just 1 to 10 micrometers, and it holds its single circular DNA molecule loosely in a region called the nucleoid. The eukaryote, on the other hand, houses multiple linear DNA molecules safely locked inside a true, enclosed nucleus. And notice that physical scale. Eukaryotes range from 10 to 100 micrometers. That is a massive 10 times difference in size.
Now, what's really interesting is seeing the sheer visual difference in size and complexity. A simple bacterium is highly efficient, featuring a protective capsule, an outer cell wall, and a whip-like flagellum for movement. But compare that to a eukaryotic cell. It's almost like a sprawling, futuristic city. It features an incredibly complex biomembrane system, specialized organelles like mitochondria, and a highly developed skeleton, which is this web-like network that gives the massive cell its internal structure and creates these little transport highways.
Section 5. Mastering essential key terms. Let's completely shift our energy now. We're going to lock in some essential vocabulary, moving from basic bacterial structures all the way up to complex human biology. First up, peptidoglycan. We actually mentioned this a minute ago as a unique identifier for bacteria. Think of peptidoglycan as the armor of the bacterial cell. Its primary job is structural protection, ensuring the cell maintains its specific shape while actively defending it from environmental stress.
Next, we have plasmids. Unlike the main chromosomal DNA and the nucleoid, plasmids are kind of like bonus genetic material. They're just small, circular DNA molecules floating freely in the cytoplasm of bacteria. The big keyword to associate with plasmids is conjugation. This is the incredibly cool process where bacteria actually swap these plasmids with one another, sharing advantageous traits like antibiotic resistance. Now for the ribosome. Whether we're talking about a super simple prokaryote or a highly complex eukaryote, ribosomes are universal. They are the fundamental manufacturing plants of the cell. Their entire purpose is to read genetic instructions during a process called translation and then synthesize the proteins that literally keep the cell alive.
Moving up to complex life, we have homeostasis. When you have multicellular eukaryotes like, say, a human being, you're dealing with massive networks of billions of cells that all have to function perfectly together. Homeostasis is the strict, continuous regulation required to keep that internal environment perfectly balanced, constantly managing everything from your temperature to your pH levels.
Finally, apoptosis. This is programmed cell death. Now, I know it sounds a bit destructive, but it's actually absolutely vital for life. While cell proliferation is the creation of new cells, apoptosis is the scheduled, orderly removal of old or damaged ones. Cells literally must die on schedule for the larger, complex organism to live healthily and avoid diseases like cancer.
So to wrap up our explainer today, I want to leave you with a thought that goes a bit beyond just passing your exam. We learned earlier that biology's epic origin story traces back to extremophiles surviving in boiling, highly toxic hydrothermal vents. Well, if life can spark in such a hostile, violent environment right here on Earth, what other extreme, seemingly impossible environments out there in our universe might currently be harboring the exact same kind of origin story? Keep that curiosity alive as you continue studying. Good luck on your exams and keep learning.